So I just want to, I'll be as quick as I can. There's some slides that I can quickly skip over. So I was going to give a bit of a brief review of genomic aberrations in lung cancer and a brief history of lung fusion testing in the Guy's lab where I work. Tissue types and challenges, specific deparaffinatation times. We've heard a lot from people around the technical challenges of deparaffination. We probably take a slightly different approach in our lab than most labs. And then I just was going to run through the ALK, ROS, RET, and then the MET copy number analysis as well in lung cancer. I've just listed here the probes that we actually use in our lab. So we use the VISIS ALK probe, the Cytovision ROS1 probe, the Cytocell RET probe, and the Leica MET copy number probe. So just thinking about lung cancer, it's quite different to the talks that we've had so far. So we already know that the patient's got lung cancer. So what we're looking for are genomic aberrations that will help determine some patient-specific treatment. So you've got a lot of patients with EGFR and KRAS variants, and what we're looking at by FISH, or typically have looked at by FISH, are the fusions, so ALK, ROS, and RET, and then MET copy number. So we're looking at a very small percentage, or we're looking for a needle in a haystack a lot of the time, but it is really important for patients that we can pick up these fusions because they're really targetable by the tyrosine kinase inhibitors. So crizotinib was FDA approved in 2011 with the VISIS probe as the companion diagnostic. There was very clear guidance around how you score the FISH probes for ALK, and we did a very big validation project back in 2011 and looked at a lot of cases that had been sent to an external provider for FISH. Our cytogenetic team were trained in the identification of lung tumour cells because they can look very different according to the subtypes of adeno. And in 2012, we went live with ALK-FISH, shortly followed by ROS1. And again, we took quite a different approach to FISH testing in our lab. We did ALK and ROS1 up front and no histopathology, no IHC for quite a long time because our histopathology team were very, very short of resources. They didn't have the resources to be able to do the IHC for ALK and ROS1, but we kind of swapped places in 2020. So we became quite short-staffed. The histo team were very much better staffed, so it seemed sensible to move over to the approach where we screen by IHC for ALK and ROS1, and then we confirm by FISH for any equivocal or positive cases. The correlation between IHC and FISH is very good for ALK, less so for ROS1. And that is kind of really clear these days when we're doing much less FISH testing because we're doing a lot of NGS testing. Had a look at our cases that we've received for ALK this year. All but one of them are positive, but none of the ROS1s that we've received are positive. So there's a big difference in the way that the IHC works. So when we moved to IHC screening for ALK and ROS, we replaced that FISH testing with RET because there isn't a reliable IHC test for RET. And then in 2021, as part of the GLH target operating model, we started to consolidate lung testing to our lab for the whole of our southeast region. So we started with lung cancers because they were the main biggest group of tumours we were testing, and we moved on to an RNA fusion testing strategy for lung. So we've got an RNA fusion panel. Why do we still need to do FISH for lung? So RNA analysis, you've got you can get a really good determination of the fusions, and you're looking at, you can detect fusions that are not part of what you would see when you're just looking at FISH. So you can find incidental findings as well. DNA copy number analysis, but the trouble is with RNA, it's a bit difficult with FFPE. Our big panel needs a lot of RNA. We get quite a high failure rate, although it's much better recently. Whereas with fish, you can do your IHC, do your fish, and that can be quite quick. Your fish probes are partner agnostic, so if you've got a break apart probe, you can say that you've got a rearrangement. We've got a really good success rate with our fish probes, and a competent analyst can reliably detect the rearrangements. But correlation with an H&E slide or a chemistry slide is absolutely essential for lung cancers, because you need to be able to be confident that you are looking at the tumour cells. So fish is also suitable for low tumour content, and providing you can reliably detect your tumour on your fish slide, you can report confidently. So the types of tissue that we receive for fish are highly variable. So we can get really big sections from lung surgical resections. We can get needle core biopsies, sometimes that are absolutely tiny. We've got EBUS, which can have, or other cytological samples that have a lot of red cells in there. So sometimes it's really hard to see the tumour cells through all the glow of the red cells. The tumour cells can be in clumps in cytology samples, and they're really easy to find. But then if you've got a very diffuse infiltration in your cell block and you've just got random scattering of tumour cells, then that can be really difficult and really challenging. And then you've got NECAS slides, which can also be challenging because they're quite different to most of the other cells that we've got. So we have a light microscope next to our fluorescent microscope, and we insist that we have either an H&E or an immunostain slide, and we won't analyse it until we've got that. So our referral centres are very good at sending us those slides with the fish slides. So I'm not going to go through this in detail, but this just kind of demonstrates how we approach the deparaffинation and the tissue processing for fish. We've got a wide variety of different times, so solution 1 and solution 2 times for lots of different types of sections, and particularly ALK is very sensitive to overdigestion. So if we've got a small tissue section and we need to do ALK fish, we use really short times for that tissue processing. So this is not a lung cancer, but it's just to demonstrate how important it is that you have got an H&E or an immunostain slide, so that you can be confident that you're looking at the right cells, because if you're not looking at the right cells, you're going to give a wrong result, and that's going to have quite a significant impact for the patient. So as Gavin has already kind of very eloquently described, ALK, the most common ALK partner is the EML4, and that is a inversion on chromosome 2. They've got 2 signal patterns with the ALK fish probe, which are very clearly defined by the probe manufacturer. So you've either got your fusion red-green, and you can have multiple fusions in multiple copies, or you've got your fusion red, and both of those are classed as positive for an ALK rearrangement. It's really important that you don't report the cells that have just got a standalone green, which is the 5' part of ALK, as being positive, which is a little bit difficult sometimes, because you see an abnormal signal pattern and you think, but why aren't we reporting this? Because it's clearly not normal, but it's not associated with fusion, because the tyrosine kinase domain is not there in that part of the signal. So we just score all of the signal patterns that we see in the 50 cells. We also score the number of fusions, and we did this because we thought at some point it was quite likely that increased copy number, particularly the high increased copy number, might also be relevant to treatment with TKIs, but that hasn't actually happened. And then we've just got a summary sheet. And then So we've got, I'm not going to go into this bit in detail because this is really clearly defined, and if you're doing ALK fish, you should be really clear on what you're looking for. So this is a typical slide with ALK fish. So you're looking at the signal patterns, and you can quite clearly see that there are some rearranged cells here. You've got big separation between your red and your green. As Paul said, you can get your finger through in between those, and you can quite happily call those cells positive. And the same with the fusions there. You can quite clearly see that they're intact fusions, easy to score those, can be confident that they're normal. But then you've got these cells here that, are they rearranged? Are they not rearranged? And I think you would not be able to clearly score some of those as being rearranged, but you would be quite suspicious if you're scanning your slide and you start seeing a lot of cells that have got a little bit of pulling apart. You need to start looking around just to see if you've got some cells that are very clearly rearranged. This probe is, in our experience, one of the probes, or the probe, where we've had the most analytical errors, largely because it's an inversion and it can be quite difficult to score. And when we were first setting this up, we had a lot of misscoring and a lot of cases that were scored as negative that were actually positive. So it's really important that your analysts are fully trained and are you know, really clear on what the signal patterns should be for ALK. So ROS1, another break apart probe. We use the Zeta-Vision. We've used probably over the period of time all of the ROS1 probes. We had real difficulty with the Abbott probe because we just couldn't get good signal intensity, but the Zeta-Vision probe works really well. So we follow the same analytical principles for ROS1 as we do for ALK. Loss of the 3' with retention of 5' again is not considered to be rearranged. Loss of 5' with retention of 3' is considered to be rearranged, but it's not so clearly defined as it is for the ALK. And a lot of work went into the ALK because it was the companion diagnostic. So, but the Zeta-Vision clearly shows one fusion and one green as being an aberrant signal pattern. With ROS1, you can frequently get loss of one fusion, so that can be a little bit disconcerting because it looks like you've got a lot of signal cut through, but actually deletion is a common event with the ROS1. So don't be too put off by that, providing your slides are the standard thickness. And then RET. So again, we follow the same principles for screening RET as we do for ROS. So it's broadly based on ALK. And RET rearrangement has also been reported as a resistant mechanism for patients that have been treated with EGFR targeted tyrosine kinase inhibitors. But in 2021, there was a publication by a collaboration between the Netherlands, a couple of groups in the Netherlands and Irish group, calling for caution with RET fish analysis, because they did a study and they found that there was a large discrepancy between what was scored as positive by fish and what was actually positive with a fusion detected by NGS. So they were saying that really, if you find a RET rearrangement by fish, you should get that confirmed by an orthogonal method, preferably by NGS. That's quite difficult for us in our lab because we use RET fish now as a salvage pathway, so the RNA has already failed, so it would be quite difficult to confirm that. But there was also a letter that was commenting on this publication and saying, yeah, okay, I've read your article, but what's really important is understanding your probe set. Some of the RET probes have got a really big gap in between the red and the green. So you could If you're just saying, right, 15% of my tumour cells, which is what you would say for ALK, if they've got a pulled apart signal pattern, then yeah, I'm saying that's rearranged. But actually, you need to do a really good validation for your FISH probe so that you're confident on what your signal patterns look like, particularly if your FISH probes with break apart have got a big gap in between them. And then MET. So MET amplification is a known resistance mechanism for patients that have been treated with EGFR TKIs. So it's a probe that's got MET in red and then the 7 centromere in green. So it's quite similar to scoring HER2 in breast cancer. We can copy it, counting the number of red signals, which is MET, against the green signals, which is the 7 centromere. In a minimum of 10 cells from each of 3 different areas of tumour. And then the results calculated both in a ratio and in a copy number. The difference between RET and HER2 is that we don't have the recommendations, the UK recommendations that there are for breast cancer. So there are as many different studies calling things into different categories as the, so it's quite difficult to say with certainty what is classing as an amplification and what is polysomy, because from the FISH analysis that we do with the other probes, we know that lung cancer, you get a lot of gains. So with MET, on the left-hand side image, you can see that there's polysomy. You've got a gain of red and green, and then there are a few extra reds in there, probably going to come out somewhere around 5 as an average across the cell. Does that really constitute the same thing as the image on the right-hand side, where you've got very clear amplification of MET versus the 7 centromere? And then this patient, this is one that we looked at recently. So this patient has got an EGFR L858R, and they've been treated with osimertinib, but they're resistant to osimertinib. And this is the MET FISH, and you can see why. But as opposed to the previous cell, where you've got really clearly defined amplification that's separate, it's probably double minutes within that cell. This, you've probably got a HSR because you've got really big clumps of red that in some cells just look like autofluorescence, but actually once you start looking around, you can see that that is amplification. But this is hard to score accurately because you can't see clearly defined signals, but you can see that you've clearly got amplification there. So I've been through that slide already. And then there's a publication from a recent publication that is saying, actually, we could probably simplify the way that we're scoring FISH for MET FISH. So this group are recommending that when you start to do your analysis, before you start doing any counts, you screen your slide, look at whether you can see any areas of amplification. If you can't, then just find 10 cells for each analyst and score those. And if you can, then again, find 10 cells from those areas from each analyst and score those. And they actually found a really good correlation. But also they were suggesting 3 different groups of positivity. So you've got high level amplification, and in the studies, the high level amplification correlates nicely with the MGS cases that are considered to be positive. Then you've got intermediate level and then low level. And I think if we could look at getting some good recommendations for MET screening, I think having those different categories would be really useful. So that was a bit of a whistle-stop tour. Does anyone get any questions? Or?
I've got a quick question.
Yeah.
So you showed your table of your different fish processing times for different tumour types. Was that guided by manufacturers? Was that in-house troubleshooting?
It's in-house. It's something that we've done over a period, a long time. And I remember we had years ago, we did a fish FFPE workshop very similar to this. And I described that and everybody looked horrified. It's like, you can't do that. It actually works really well in our lab. So, you know, we've got all the different times and you start off with the long ones and then the ones that have got shorter times get put in the coffin jars later on. So it does work really well.
But again, that's something that you did in your setting.
Yeah.
So it might not be applicable.
No,
to
absolutely, yeah. But it's something that I think rather than saying all lungs have to have this digestion time, if you can think about some flexibility within your times, but they would need to be times that you validate yourself according to what your own pre-treatment steps are.
Yeah. Okay, good presentation. Any questions from the floor? I don't think we've had any online for this one.